I'm going to sort of talk about some of the other things that birds do that are intellectually appealing to us. They are, as he said, like us in that they're active during the day. They're one of the few groups of vertebrates or animals in general that sees color. They communicate with sound. They pair up. The dark side of it is that they unpair, and they, in some cases, they commit incest. They commit adultery and cuckoldry, and that's sort of what I study. I'm kind of fascinated by those aspects. Sue asked me to send you something to read, and I thought initially I would just talk about a bird song, but then I got greedy, and knowing that it's a group of librarians, you can actually keep two topics in your mind at once. So I'm going to talk about bird song, but I'm going to begin by letting you kind of follow what was in my mind that led to this study of bird song. And it actually began with incest avoidance. And I'll explain what I mean by that. Now, when you put color bands on birds' legs and you follow them around in the field and you see who is associating with whom, and so I know some birders who keep a separate, not just a bird list, but a copulation bird list of species that they see actually mating. And you look at individuals and who feeds the young, whose territory is the nest placed on, and you can start to pair birds up. And you can actually reconstruct their social lives the same way you could be with people, and probably the Patriot Act has a pretty good idea, thanks to that, of what you all are doing. And we're all followed around. Well, this is what we do with birds. And we can look at questions like divorce, for example, or incest. And the birds that have been best studied are these four species in terms of whether or not birds nest in the territories of their close relatives and end up mating with them. The blue tit and the great tit are members of the same family as chickadees, very closely related. Collared flycatcher would be a little more closely related to thrushes like American robins and bluebirds. And house sparrows are a representative of an old world European, Asiatic, African family of birds that are not at all closely related to our sparrows. So these are the four kind of model species where people have looked at the mating systems of birds. What do you notice about these four bird species? They're all cavity nesters, and they're in bird boxes. And when you put out bird boxes, you've already done a manipulation that potentially changes the behavior of birds. For one thing, it's a little bit like a subdivision. Every house is identical. So females don't necessarily choose males on the basis of the quality of their nest box, because they're all identical. It's just totally homogeneous. Also, bird box studies and bird box populations tend to be so highly subsidized by provision of boxes and prevention of predation by the ordinary predators that might lower population densities. So they tend to be very dense populations, which could increase laying of eggs in other birds' nests, alter their behavior in a lot of different ways. So I wanted to ask these same sorts of questions about mate choice and divorce and incest and its effect on longevity, but I wanted it in a more natural bird species. So I worked on a species called a Savannah Sparrow. Now, I should say that for my dissertation, I worked in Monteverde, Costa Rica. I bet a number of you have probably been there. If you haven't, really, you want to go. It's an absolutely lovely place. And I worked on maybe the most beautiful bird in the Western Hemisphere, the resplendent quetzal. Great, beautiful, awe-inspiring bird, but the devil to work with. It would blend away into the tropical forest. It is a bird that's highly threatened in many parts of its range, which is quite a narrow range. It's an important symbol of culturally throughout Central America. It's a bird that you really can't manipulate very much, either legally or practically. So you can't actually ask these sort of questions with this beautiful bird. You've got to find kind of a drab bird that people don't care as much about. So Savannah sparrows don't look like much, but it turns out they've got an absolutely spectacular biology once you look at them closely. So they're a bird the size of a house sparrow, which you know from, even if you're not a birder, if you go to McDonald's and see the birds that are scrabbling around for the crumbs, those, or in airports and malls and things, those are house sparrows. This is a different family, but ecologically somewhat similar size. Males and females look just the same. Males are a little bit bigger, 5 or 10% or so. This species is migratory. What else do I need to tell you? Well, I'll describe some more of the biology as we go along. Its breeding range goes from western Alaska right up into the Maritimes and New England and down into the south, and it migrates to the southern United States and into Central America. Even though it has one of the widest breeding distributions of any North American bird, probably a lot of people have never even seen a Savannah sparrow. How many people have seen a Savannah sparrow? And how many people have seen a song sparrow? So yeah, a lot more hands go up. I don't know if there are more Savannah sparrows than song sparrows, but they're probably pretty darn close. All you have to do to find a Savannah sparrow is to go into some big open field in the blue area here, and you'll find them. But they have this, as I'm going to show you in a moment, very high-pitched, not terribly melodic, kind of repetitive insect-like song, and they run around like rodents in the grass. You just don't see them, but they actually are everywhere. And where I ended up studying them is at the Bowdoin Scientific Station, which is located on an island in the Bay of Fundy called Kent Island. So it is actually almost due east of here, a little bit to the north. To get to Kent Island from where we are now in Brunswick, you have to go up. You can see Cape Cod there in the lower left, and the circle is around Kent Island. It's just south of the large island in the Bay of Fundy, and we're at the northern Gulf part of the northern part of the Gulf of Maine. The big island is called Grand Manan Island. And so to get there from Brunswick, which is maybe halfway between Cape Cod and the Red Circle, you drive 4 hours to the Canadian border at Calais, St. Stephen, drive another hour into Canada, take a 2-hour ferry south from Black's Harbor. You land at the northeast tip of Grand Manan Island. You drive to the southern village of to Seal Cove, and then you take an hour-long lobsterman's boat out into the bay, shift all your stuff into a dory, paddle to shore, and walk a 1/4 mile, and you're there. So I can get to Costa Rica actually way faster than I can get to Kent Island. But the advantage of both places that you leave, well, I won't actually. Anyway, you simplify your life. You leave administrators behind who could cost your life, and you leave actually flush toilets and electricity behind, but also email and internet access, at least when I was there. And that actually was kind of a relief for me. I've often sought out speed bumps in my life, and that was a good way to get out to Kent Island and have 2 months focusing in on something really simple that was fascinating to me, and I could test some interesting scientific questions without some of the distractions that I get on the mainland. When you arrive, actually that I think might be our lobster boat, that white dot. Do I have a way to, yeah, that work? It's not really showing, I don't think. Ooh, there we go. Yeah, that's where the boat would be moored. You would take the dory to shore here. You have a high tide that you can't cut across the basin here, so you have to walk around through the woods. And you come up here, past the Northfield. That's one of my study sites. And this is known as downtown. That's the dorm where everybody lives while they're doing research on the island. So just to kind of give you a little bit of history, I don't know if this is appropriate for a talk like this, but what the heck, you'd never guess how this island came to be the Bowdoin Scientific Station. So I'm just going to have to tell you. And I'm going to give you the very, very quick version of it. In the late 20s, a local fisherman on Grand Manan named Ernest Joy saw a bird he'd never seen before. And Michael kind of made allusions, I think, to how ornithology used to be done. So he did what most ornithologists of the era would do, which is to shoot it. And then he said, whoa, what the heck is this? Gave it to another Grand Mananer named Alan Moses, who made a specimen of it. And it turned out to be a yellow-nosed albatross from the Antarctic. What it was doing in the wrong hemisphere. It was the first recorded record of this species in North America. And the American Museum of Natural History in New York got wind of this, and they wanted the specimen, of course, for their collection. So they sent a delegation up to Grand Manan with some mirrors and beads and things like that to trade for this specimen. But Alan Moses was a little smarter than that, and he said, no, you can have the specimen, but on one condition. I'd like to go on one of your expeditions somewhere. I've never been off Grand Manan. Well, several years later, he found himself in the Belgian Congo with J. Sterling Rockefeller, one of the world's wealthiest men. And they were in search of a bird called a Grauer's broadbill. See, I told you never would have guessed this. A very rare, small African species that they also wanted to add to their collection. And they couldn't find the bird, literally weeks of tramping through the rainforest of the Belgian Congo, unsuccessfully looking for Grauer's broadbill. And Alan Moses caught malaria, so he couldn't even tromp anymore. And they left him in a hammock in the woods while they went off to tromp again to find it. And in his malarial, feverish stupor, all alone in the camp, he looked up on a branch, and there was Grauer's broadbill. And he shot it, of course. And the bird fell down, and he grasped it and then collapsed again. And they came back at the end of the day, the rest of the expedition, and they had, of course, had another fruitless day. But there's Grauer's broadbill on Alan Moses' chest. And J. Sterling Rockefeller was so grateful to him. He said, "What can I do, my good man, to pay you back?" And he said, "Well, back at Grand Manan, there's a little island which is a refuge for a species of duck, common eider, now a very common bird in the Gulf of Maine, but back then it was down to about 30 individuals. And they bred on Kent Island, and he said, protect that island." So that happened in 1930, and J. Sterling Rockefeller bought the island for a small amount, talked to Ernst Mayr at Harvard and said, "What do I do with this island I now own in the Bay of Fundy?" And he said, "Give it to Bowdoin College as a research station." Complicated little story. It's a very special island with a special history, and many Bowdoin students and many graduate students from various other institutions, various faculty from around Canada and the United States have done research there for the last 70 or 80 years now. Not only do you not get pestered by sort of modern life while you're out there, there's no mosquitoes, there are no black flies, there are no deer ticks. So it's just a place that you don't have to worry about a thing, except for one thing. About 1 in 100 of the herring gulls that nest in our study area is pathological, and they will actually dive bomb you, and so you have to wear a hard hat if you're looking for Savannah Sparrow nests in their territory. The way they do it, they'll go into a stoop just like a falcon, and at the last moment they'll lower their legs and they'll rake their middle toe right across your head. And that's what that one looks like. So within the study site, we have 100% of the Savannah sparrows uniquely banded. And they're banded in two ways. They have, you've probably seen, I should have brought some in to pass around, but aluminum U.S. Fish and Wildlife Service or Canadian Wildlife Service bands that have a unique 9 or 10-digit number. So if that bird then leaves Kent Island and a cat kills it in South Carolina, it can be brought into a local authority or it can be sent into the bird banding lab, and they will have a record of that band. We'll know exactly the date it was banded, the date it was killed, how long it had traveled, how old the bird was, and that's really how, until more recent techniques, that's how we reconstructed birds' migratory routes and so on. So every bird has a permanent metal band on its leg, but it also has, in our study site, as I'll show in just a moment, 3 randomly assigned plastic color bands, and that allows us at a distance of 100 meters to identify each individual. In my study area, it's gridded, just using a lawnmower basically, into 50-meter on a side squares. So we try to walk on the paths because the birds nest in the grass, but from the edge of any one of these squares, you can scan across the square and see with your binoculars who's who, what their behavior is, who's copulating with whom, where their nest is, who they're feeding in terms of nestlings, and other sorts of interactions. The white spots, they may not show up very well, are tree swallow boxes, which is another long-term population study that we've been working on. So we put up, to capture the birds, these mist nets, and we have competitions to see how many we can get before breakfast sometime when they start to flock later in the season. And I think my record day was 80 birds banded before breakfast, all Savannah sparrows. And this is a net, just looks like kind of a badminton net, pretty much, but of the material that your grandmother sort of head nets might have been made of. Just a very fine filament, and the birds don't see it, and they collide into the net and fall into kind of a pocket. And then it takes a little bit of practice to learn how to get them safely out of the net. But once you do, this is how you adorn them. Now, I mentioned that we randomly assign color combinations to these birds, so you have no two birds alive at the same time with the same color combination. So this bird I would call L.SR, and that would be light blue over aluminum on the left leg and stripe over red on the right leg. So if I pop my binoculars up, oh, there's L.SR, I can see him, and he's fighting with B.NY. One of the reasons that you have to randomly put colors on was revealed to me, fortunately, right at the beginning of this study. I'd had a plan that I was going to just take all my red bands with all the red combinations, red plus blue, red plus white, red plus yellow, kind of motor through those in a systematic, scientific way, and then move to the next color. But just at that time, Nancy Burley came out with a publication showing that females really love certain colors. And it turns out that they're crazy, for example, about red color bands. So I could have taken all the birds in one of my study areas, banded them all red, found that all the females go to mate with those males, and had essentially something that could have confounded what was really going on. When we capture the birds, as in, this is very common to most field studies of ornithology, I'm sure, we take various measures of size, how big the bird is, and the best ones are weight, wing length, the length of their tarsus. We measure their bill, like Peter and Rosemary Grant's well-known work on Galapagos finches, because bills are so important for feeding behavior. We take a blood sample now often to know something about paternity using various molecular techniques. You might measure hormones in the blood. We might make a sample of a feather. So we're measuring a whole bunch of things about the birds as well. So we have, on each one of these birds, I have a database just like the IRS. I've got probably, 30 or more variables associated with each bird. So when I see it, I can get on the computer and see its age, where it was born, if I know that, who its mother and father are, all of its nests, and so on. So then, with my field assistants, we sit and record. You don't recognize anybody here? Lower left? Remember him? Well, you get another shot at his brother maybe this year. So we spend a lot of our time just quantifying where the birds are, monitoring the birds. Yeah, he's got the lens caps on this. I don't know what that's Okay, let's see how good you are at finding Savannah Sparrow nests. I do this test with my field assistants when I'm trying to choose who's going to help with the Savannah Sparrow project, or whether I should have them work on irises instead. Have you found the nest yet? Yes. There it is. Yeah, it can be. It's maddeningly hard to find these nests. That's why people work on bird box studies, because it's easy to find the bird box nest. These things are hard. If I'm really on a roll, I can find a nest every hour, but that's a good day. Sometimes it can take you 5-6 hours to find a nest. But then we monitor it for egg laying, for hatching, for predation. When the young are 8 days old and ready to leave the nest, we'll take the same kind of measurements we did with their parents, put permanent bands on them as well, collect a blood sample, a feather or something like that. And now we have information on the date of hatching, the date of fledging success, the quality of the offspring. Are they heavy? Are they underweight? How many of the nestlings failed to fledge? Early on, we discovered, we knew a lot of this from the literature, but we had some banding recaptures from some of the birds that we had banded, and we now know that Savannah sparrows that breed on Kent Island actually fly 1,600 kilometers down to the southeastern United States. Actually, they're probably spread from northern Florida, we know that actually, from northern Florida right up to, say, Virginia or so. But a great long migration. Here is the incredibly fortunate aspect of their biology in terms of allowing me to ask the questions that I've occupied myself with over the last 20 years. They come right back to where they were born. So here's an example of what's called natal philipatry. So philipatry just means love of your homeland. And it refers to the fact that you come back to the same spot. And you can come back as a breeder, so that's known as breeding philipatry. If in your backyards, if you've got American robins, next year there's a pretty good chance that the robin in your, well, there's a 50% chance that the robin in your backyard is the same robin. And that would be an example of breeding philipatry. They come back to the same site. But it's very unusual to have what's called natal philipatry. If you were to band all of the baby robins in your backyard, you'd never see one again as an adult because they would be 100 miles away, 400 miles away, 10 miles away, but you're not going to see it. But these Savannah sparrows show extraordinary natal philipatry. Perhaps not surprisingly, because if you aren't pretty precise in returning and you're like off by 5 miles, you're in the Bay of Fundy. So they tend to come right to the same site. So here are two examples. If you look at the green and the yellow triangles, first with the green one, it was born where the triangle is, and then it had a whole succession of different nests over the years. But it came right back to Kent Island. It dispersed from its natal site only a couple hundred meters, which is nothing. And then each breeding site is only on average about 30 meters away from the previous year. So extraordinary breeding philipatry. Here's another example to show the same thing. So what that allowed me to do now was to say, I remember you when you were an egg. And I knew your mother and your father. I knew your grandfather and your great-grandfather and your great-great-grandfather. And so we put together this pedigree. So this is a family tree, just what you've always wanted to see, of Savannah sparrows on Kent Island over a 12-generation period. I'm not going to talk about it today, but this has allowed us to do work in quantitative genetics to estimate the heritability of any trait that you might be interested in, not just body size, but also the heritability, the likelihood that you would inherit a gene for divorce, or more properly, a whole suite of genes that predisposed you for not staying with the same mate, or that you would be polygamous, or that you would live a long life or a short life. So this is something you can only do in a natural population if you have natal philipatry. And I had no idea when I started the study that I'd be able to do that. So finally, we're at incest avoidance. Now, is incest that, and by incest I mean breeding with a genetically close relative like a sibling or an uncle or a grandson, is that a risk in natural populations? Well, we don't know for most natural populations because they're not natally philipatric. We don't have these pedigrees, but we do with the Kent Island population. So here's just one example. Again, focus in the bottom at the green triangle. That's a nest. There was a female at the nest, a male at the nest, and some offspring. And the following year, the mother moved about 50 meters to the north. So she showed breeding philipatry. She didn't go very far, in other words. It's not as if she went to Nova Scotia or Maine. She stayed right on Kent Island, moved 50 meters to the north, but she divorced the male, so he now had a different nest. He was over here. One of the females from that nest, their daughter, moved not very far, given that she had migrated all the way down to the Carolinas and then come back and found the island, and then the son was here. So if you're a particular female, take that mother, you could mate with, take the daughter would be a better example. There are two possible incestuous matings that she could have had. She could have mated with her brother or with her father. And we repeated this for literally thousands of pairs, and what we noticed early on in the study is that we weren't seeing incest, even though it was a minefield of potential incest. Relatives are all over the place, and I'm not even showing you uncles and grandfathers and so on. So we published a paper and in which we showed that inbreeding or incest was extraordinarily rare. This is about halfway through the study. Now it's interesting, some of the text is not carried over on this presentation, but the very year that we published that study in the journal Ecology, another paper came out on a related species of sparrow, and the title of that paper was no evidence for inbreeding avoidance in a natural population of song sparrows. So we found inbreeding avoidance. They claimed they found none. And there was a second paper a couple of years later, also entitled No Evidence for Inbreeding Avoidance in a Great Reed Warbler Population. So who's right? Is there inbreeding avoidance in Savannah sparrows, but not in song sparrows and great reed warblers? Did we do something wrong? Did they do something wrong? Did they overlook something? So that became kind of the next step in this study. So I want you to think for a moment about incest and about genetic relatedness between you and your relatives. So focus here on the female, that's the red on the left. And you can calculate as a geneticist something called the coefficient of relatedness. And all it is really is the proportion of genes that are identical by descent that you share with somebody. So what proportion of the genes that you have in your body do you share with your father? 50% came from your father, 50 came from your mother. What proportion do you share with your brother? You have exactly 50% of your genes from your father. You have, on average, 50% with your brother. You could have more than that, you could have less than that, depending on meiosis. And what proportion do you have with your son? You have exactly 50% in common. So that's the coefficient of relatedness. Now I have little asterisks by the father and the brother, and the reason I do is that birds have rather leaky mating systems. There's a high degree of what's called extra-pair paternity. And that is that the male who thinks he's the father of a particular bird, depending upon the species, sort of 5 to 50% of the time, he's not actually the genetic father, that a neighboring male is the true genetic father. So you actually would want to discount that 0.5. You're probably a little less likely to share genes with the male that you think is your father. And the same is true for that reason with your brother. He may not be your brother. He may have been sired by someone else. So the main point I wanted to leave you with this, I'm building up a model about where you should expect to see incest, and in particular why there should be some asymmetry. And the asymmetry doesn't originate here really, because a female mating with her father in terms of the genetic risk of inbreeding, or with her brother or with her son, should be roughly equal. Everybody with me on that? You share, on average, discount, not talking about extra pair paternity for the moment, but on average you share the same amount of genes with each of them, so you should expect natural selection to favor avoiding inbreeding because of all the deleterious problems with inbreeding equally in all of those relationships. But for a number of reasons, we argued it should be asymmetrical. And we had two main hypotheses. One we called the incomplete knowledge hypothesis. What you know about your father or your brother or your son is different. So you're a young female in a nest. You're just hatched out of the egg. Do you know your father's song? That's a question for you. How come? Because you listen. In fact, he makes it easy for you because every time he comes down to deliver food to you, he jumps on a perch, and he sings, and he's basically saying, "Ladies, did you see what I just did?" So every time you get food, you hear his song. And that's actually a pretty powerful mechanism for learning. So you know your father's song. How about your brother's song? How would you know your brother's song? Because what? Yeah, but he's a little nestling. He won't even begin to sing for about 6 months. You're in South Carolina. He's in Virginia.
Does he learn the father's song?
Great question. That's why so that's the second part of the song. That's why I needed to figure out where do they learn their songs. But so if he learns their father's song, not a problem, because you know the father's song. If he doesn't learn the father's song, you don't know what your brother's song sounds like. You don't know what your son's song sounds like for the same reasons, because you last knew him before he ever sang. So you know your father's song, you have knowledge about your relatedness to him, at least your social relatedness. Now, he may not be your genetic father, as it turns out, but you got a pretty good guess that guy is the one who thinks he's your father anyway. You don't know who your son is. So which one are you more likely to accidentally breed with? Your brother or your father? Brother. Good. You guys are all here. Okay. The second one is the cost of inbreeding. And the behavior of animals is really like an economic set of decisions. The currency is not dollars and cents, but fitness, essentially, your lifetime reproductive success. And avoiding incest can be costly to birds if they have to move their territory in order to escape an insistent a relative who's courting them insistently. For females, it may mean actually giving up their breeding philipatry and going somewhere else to get away because there's a risk of inbreeding. That's a cost of inbreeding avoidance. The costs of inbreeding themselves are likely to be asymmetrical, we argued. So we're sort of developing a series of hypotheses. Michael talked about that early on, but it was based on lots of observations in advance. The costs of inbreeding, would you imagine they're higher for males or for females? Who should be more discriminating? Why? Because they put more energy in and more time and take more risk caring for the young. A male Savannah Sparrow can go from one female, copulate with her, go to another, bang, 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 and it really doesn't matter. Relatives, not a problem. He would have lower reproductive success with a close relative, but if he's not doing the work and it's sort of extra, natural selection would not penalize that behavior as much, whereas natural selection, we argued, would penalize a female mating with a male who's a close relative because her offspring would have lower success because they would be highly inbred. So now you can see there are asymmetries of age and of sex. So our prediction was, just as you said, we would expect incest to be very rare between females and their fathers, but it should always be uncommon, but brothers and sons not as rare as fathers. So what did we find? So here I'm distinguishing social incest from genetic incest. Social incest is just, I've got my binoculars out, I read your color band, you're both hanging out, I'm going to call you a pair, you're acting like a pair, you're both feeding the young, so that sort of is more evidence that you're a pair, you're at least a social pair. So that's how we determine whether it's incestual or not in terms of their social behavior. And what we found over, 17 years, 1,100 pairs. This is not for the faint-hearted. You got to like Savannah Sparrows to do all that work. But we actually found 3 mother-son pairs, 6 sister-brother pairs, and 0 father-daughter pairs. Now, overall, the probability of incest was still far lower than you would expect by chance. And you can prove that with, or at least you can provide evidence for that statement, by doing kind of a computer dating game, which is what we did, a Monte Carlo simulation. And you take a female who's alive, and you randomly go grab her, a living male, and you pair them. And you just do that 10,000 times, and you say, how many times if I randomly grab male and female do I get an incestuous pair? And it turns out we should have had 138, we got 9. So that's evidence they're using some cue to avoid incest. That's social. In 3 years, we actually did the work of collecting blood samples and looking at paternity analysis through molecular techniques. And this is structured in a slightly different way, but the main point is we got no offspring genetically from father-daughter pairs, and very, very few in any other types of pairs. So neither socially nor genetically are they choosing mates randomly with respect to relatedness. So that's cool. Who knew that sparrows can recognize their relatives? That's what this shows. This was kind of neat too. The ages of incestuous individuals are non-random. The very few males that we found that were incestuous were all young males. They'd never bred before. Probably males that were having a difficulty finding a territory because all the territories are held by dominant previously established males. So these males are wandering around trying to find some place to get in. Essentially, they're not very picky. If it's a sister or a mother, not their problem. The very few cases that we had of females who were incestuous were old females. And our interpretation here is, as they approach the end of their lives, now birds are said to have age-independent mortality, so they have 50% mortality every year. But in fact, with longer term studies, we now know that they do have an upper limit to how long they live. The older they get, essentially their standards drop a bit. And if the only males around is my son, I guess we'll deal with that. Okay, so this then raised the question of what in the world are they using to avoid incest? And so we thought maybe it's song. Because if song were inherited either genetically or through learning, then that could be the cue that females are using. Because otherwise, you can't tell these things apart. Visually, they look just the same. And so a bird could learn his song, and only males sing, by the way, in this species and most temperate zone species. He could learn his song from really one of 5, possibly 6 groups. He could learn from his social father. That's the bird who's behaviorally mated to his mother. And we know the mothers are the real mothers. The social mother is the genetic mother. Females of this species don't go laying their eggs in other birds' nests. So he hatched, he knows who his mother is, she knows who her son is. He thinks he knows who his father is, but he can't be quite sure. He could have gotten his song from his genetic father, and that would be a neighboring male, in which case there would probably be a gene or a set of genes more likely that produced different songs. Or he could have learned it when he was a nestling or a fledgling, quite young that summer, from some neighboring adults. Or 4 and 5, he could learn it not this first year, but his second year, when he came back to Kent Island because of natal philopatry as a breeder, in which case he could eavesdrop on his older, more experienced breeding year neighbors, or possibly from his peers. So we can actually test all of those because of this system, which is wonderful for a field study that's not a bird box study. One thing I can't do is to know who birds listened on their wintering grounds, because they could have heard anybody from South Carolina north. So we really don't know that. We kind of finessed that. I need to show you something about sonograms and how to read this way of quantifying bird songs. And maybe some of you know this already, and it's not just bird songs, but it's noises in general. So it's read just like a musical score from left to right. So that's time on the x-axis, and on the y-axis, which says frequency in kilohertz, that's just pitch, just how, whether it's a high note or a low note. So you can see this particular song starts with short bursts. I'm looking now at the middle graph here. Short bursts of high-pitched notes, and then it gets a little more complicated and ends on some lower frequency notes. The oscillogram at the very top, A, is how loud it is. So you can see this one, well, I'll play you some in a moment. What I want you to see, because this will help you read some of the next sonograms, is that the song of a Savannah Sparrow, this is a fairly typical Savannah Sparrow song from this dialect, this northeastern dialect, has 4 parts to it. So see if you can pull out these patterns. It begins with a series of these high-pitched introductory A notes, as we call them, and they're actually interspersed with those vertical clicking sounds. So that's the introduction. And that's kind of like bum, 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 bum. That's like something is about to happen. It's an introduction to some really interesting information that's about to be produced. The middle section, which is number 2 here, has, in this case, some high notes and ch notes and a dash. That's the signature of a bird's individual identity. That almost always varies between individuals, not the introduction. The introduction says, I'm a Savannah Sparrow, I'm about to tell you who I am. And then middle tells you who I am. Then there's a little intermission, the buzz, and then the terminal trill is, in case you didn't know who I am, this is who I am again. So that combination of the middle and the terminal trill are really important. Okay, so here I have to go out of this view to play it. So I'm going to show you 3 songs of different males, and the N.WN is navy on the left leg, white over navy on the right leg, and so on. So these are 3 different males. Here's what their song sounds like. Ready? I don't know if I can get that volume up a touch here. Let me try this. Let me play that again. I think I'm good. I'm going to slow that down. Can everybody follow that, or should I show you what that's like? So, okay. So that's a pretty standard one. This one, hopefully your eyes can see the difference in the middle. That's what I mean about it being a signature of an individual. And I'll play that. And then this one we call it has sort of a stutter. So they're all Savannah Sparrows, obviously, but each one is different. And with a 10 minutes training, you can train yourself to recognize not just what species is singing, but who is singing. It's Bob, it's Fred. Oops. Okay, well, there we go. The second thing we discovered, besides this individual variation, which you can either hear or see on a sonogram, is that males sing the same song and only one song their whole life. So it gets pretty boring. They probably sing, 10,000, 100,000 times over the course of one breeding season, and then they come back and do the same thing the next year as long as they live. And here's a bird early in the study showing this same identical song that he produced. This is the only thing he ever sang, a 2-second song. So what this tells you so far is if you're a female listening, you can tell every male in the population, they all sound different. They're reliable and consistent because they only sing one song, and so you have a way of identifying individuals. The question is, do they inherit their song from their father? So now that you know how to read sonograms, you can look at the sonogram of this bird and make a prediction in your mind what its son looks like. Actually, this happened to be a fairly lucky sample size because remember, birds have 4 offspring typically in a nest. They have to survive to fledge. They have to survive to independence. They have to fly down to wherever they're going to spend the winter. They have to survive all of that. They make it back to Kent Island. Very few of them make it back. Then they have to be a male if they're going to sing. So to actually get recordings of adult offspring is actually quite hard. You've got to get many, many broods before you luck out. But we lucked out here. What do you think the sonogram's going to look like? Just like that? Is it like its father? Not very much. The middle's pretty different. Yeah. So here's a case where the middle part is really quite different. And here's another son from that same nest. And we know that those 2 sons had the identical acoustic environment. That is, they were brought up hearing the exact same sounds, and yet they, as adults, produce different songs. So this gets back to your question, do they learn from their father? This says no. They're picking up their notes from neighbors or from various other individuals. And in fact, we were able to kind of break it down. And it turned out that these are 2 different ways that we quantified similarity of a male song with potential tutors. One was using a digital sound analysis program, and the other was just visual eyeballing, where we had 3 ornithologists look at these sonograms and rate how similar they were. And we got pretty similar kinds of results from the quantitative and the more qualitative. But the important point is it wasn't that common for males to sing a song like their father. And we had no cases where they sang a song like their genetic father, which would indicate that there's unlikely to be a genetic basis for variation in song. Most of the time it was a hatching year adult male or some male the following year. One of the neat things, this is really now getting off the topic of incest, but one of the outcomes of this improvisation and opportunistically taking notes from different individuals that you hear and compiling them into your own song is that songs, like language, show cultural evolution. So in college I used to say, far out and groovy and things like that. And you don't hear that anymore. Now everything's awesome, and the same is true with Savannah Sparrows. I've really watched over the years how their songs have changed. It's kind of neat within a population. One thing in particular is that the trills at the very end have gotten shorter and shorter. These parts just emphasize what's different in this particular lineage. But here's an example of the trills. Ordinarily, back when I started this study, the birds sang like this: seet, 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 dee, 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 and now they go seet, 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 dee, dee, chirp. I kind of liked it better in the 80s, but so to go full circle, this was motivated just exactly like Mike was talking about by simple curiosity. I can't believe that I've gotten paid to go out and watch birds and think about their lives, and it's just generated dozens of questions every single day I've ever looked at them. And the search for trying to understand how they recognize their kin led me to a study of song. I haven't answered the question of how they are able to avoid mating with kin. If I had to guess, it must be something in the ultraviolet that I don't see in terms of reflection of their plumage. For all I know, it says the Smith family across the top in UV, or it's something equivalent. It may be pheromones. I don't know. Michael, you have an idea?
I like the UV.
You like the UV, okay. It could be. Still don't know, but that kind of keeps us in business, so I get to keep going back and thinking about it.